Hello and welcome to the Sigma Psi Distinguished Lectureship. I'm Katie Burke, Digital Features Editor at American Scientist, and I'm speaking today with Nick Hudd. He's a biochemist at Georgia Tech who studies the molecules that led to the origin of life. Nick, thanks for joining us.
Oh, it's great to be here.
So just a couple of notes for those who are watching the Google Hangout live. You can submit questions for Nick on the right side of your screen. If you don't see a spot there, you can also type them on the event page or on Twitter using the hashtag AmSciGHO. Okay, so let's start with a little bit of background info. Nick, what's the timeline of the origin of life, and how have scientists in the past approached understanding the origin of life and early evolution?
So I'd say the timeline is set by two very different pieces of information. We think that life must have been up and running, so to speak, at about 3.5 billion years ago. And that is because we have what are pretty credible microfossils of single-cell organisms from around that time. We also have evidence, say, carbon deposits and such that correlate very much with the presence of life. So we know that life is at least that old. We don't think life is older than 4 billion years ago, and that's because we know that based upon the craters on the moon, that around 4 billion years ago, there was quite heavy bombardment of the moon and therefore the Earth as well by asteroids. And so although there's some debate about when exactly that bombardment would have ceased or dropped to a level where life could have existed, it's generally thought that around 4 billion years ago, or maybe a little longer ago, that the impacts were so severe that they would have sterilized the Earth, even if life got started before that.
And so what did some of the scientists who started talking about the origin of life, Darwin being one of the ones that is often cited, what did they say about where they thought life came from?
So Darwin's kind of a special case in that he was so insightful. And we know that from a letter to one of his friends, he imagined, he was the one who coined the term a warm little pond with all sorts of phosphates and ammonia. And he speculated that maybe that these molecules could come together and make some of the molecules that are necessary for life. But actually, when you look at writings around his era, around the turn from the 19th to the 20th century, scientists were thinking that life started with single-cell organisms. They were thinking that, but they thought that it seems like the more widely accepted opinion was that life was starting with cyanobacteria, that is, bacteria that could harvest sunlight and make the nutrients they needed. The idea being that they would be essentially the smallest self-sufficient organism. But that really changed in the 1920s when two people, one of them being Alexander Oparin, who was in the Soviet Union at that time, and J.B. Haldane, who was in the UK. But at the same time, they proposed that maybe life started with simple organisms that were not able to harvest sunlight, were not able to manufacture their own nutrients, but were really living off of molecules that were already on the Earth, and so that they could be so simple that they could get by with the ingredients that were already present.
Okay. So then when we start getting into earlier time periods of before there were actual living organisms, what are the molecules that have gotten the most attention and debate, and what has been the accepted story since about the 50s?
So I'd say that because in the early 1950s, Stanley Miller found amino acids in his reactions, when he was modeling the atmosphere of the prebiotic Earth. There's been a lot of investigation into amino acids and how peptides would have come about. That is, amino acids stitched together. And so
and amino acids are the building blocks of proteins. So
yes,
like the
exactly.
protein in our diet and in our bodies. Right.
Exactly. And what's important about the experiments that Miller did really was that we pretty much make all of our proteins out of 20 amino acids. Yet there are many. There's essentially there's an innumerable number of possible amino acids. We only use 20 in living organisms for our proteins. And when Miller did his reactions, I think in his first experiments, he saw 3 of those that are in the 20, which is pretty amazing because, again, there's so many possibilities. He saw some other molecules that we don't find in life and some other amino acids that aren't used in proteins. But the fact that he produced amino acids that are found in our proteins was really an important advance. People were thinking about that for many years, I'd say. And then the focus shifted to RNA. There were some, and RNA is a close cousin, you might say, to DNA. It's an information storing molecule. And the focus...
It stores our genetic code in some organisms, yeah.
Exactly. So, well, DNA stores our genetic code, and RNA plays a big role in that it takes that code and it uses it to make proteins. And there's some viruses that store their genomes in RNA, but most of genes are stored in DNA. But there were a few scientists that proposed that maybe RNA came before proteins, maybe it was earlier. And then in the early 1980s, there were 2 discoveries, one at University of Colorado underneath in the laboratory of Tom Cech, and another one by Sidney Altman's lab at Yale, that discovered that RNA molecules could catalyze reactions. And until then, it was only thought that RNA and DNA were information storage or maybe scaffolding molecules. Say there's a giant complex that's called the ribosome that makes proteins. A lot of that is RNA, but they weren't thinking that these molecules could catalyze reactions. When it was found that they could catalyze reactions, then much focus went on to RNA because now you have a molecule that can both store information and catalyze reactions. And those are 2 big things that you need to get life going.
So how is RNA formed in a living organism's cells? And how does that compare to what you think could have happened on early Earth?
So that's a really good question, because there are some reactions that we see in life and we think, oh, maybe these are still carryovers or similar to those that got life started. Then there's other reactions that look very, very different from what we have in life today, or what we would think would have happened early on. So in life, the units of RNA, so RNA is a polymer, many units linked together. The single units that are linked together to make RNA have a nucleobase, we call it the pairing base, like you think about in the middle of the DNA duplex, those parts that are pairing together. And then those are connected to a sugar, which is ribose. And then ribose has a phosphate group on it. So that's like the building block of RNA. But when organisms synthesize RNA, they actually synthesize it from a triphosphate. And that is that you have the base connected to the sugar, and then instead of 1 phosphate like you have in RNA, you actually have 3 connected together on that molecule. And the reason for that is that is a high energy molecule. And the best example is, most people are familiar with the letters ATP. You know that that's an energy storing molecule and that you're burning ATP when you're exercising, right? Well, ATP is actually one of the monomers, we call it, from which RNA is synthesized, because A is one of the bases in RNA. So that's used throughout all of life, but it's a little hard to think about how that triphosphate molecule would have been made on the prebiotic Earth. And so we're thinking that maybe different reactions help stitch together the parts of RNA early on.
So returning to Miller's experiment and kind of talking a little bit about how the presence of liquid water is important to these biopolymers, the polymers used in life, and what assumptions Miller made about what the Earth's early conditions were, and what are some of the ideas you've tossed around and your colleagues have tossed around as you've looked at other possibilities?
So when it comes to Miller's experiments, a lot of people now will question whether or not the atmosphere that he had. Hold on one second. Just get a drink here. So the molecules that he had were formed from what's called a reducing atmosphere. And today we actually live in a oxidizing atmosphere. And if you try to make his molecules in an oxidizing atmosphere, it won't work. And nobody thinks that early life started in an oxidizing atmosphere. But there's a lot of questions about whether the atmosphere that Miller used is the one, because he used what we would call a strongly reducing atmosphere. And kind of the way that we classify these atmospheres, on one extreme would be strongly oxidizing, which is what we have today. We have a lot of oxygen, molecular oxygen, O2, in the atmosphere.
Which really happened when photosynthesizing organisms started.
Exactly. That is, I think, such an interesting point, which is that our atmosphere is the way it is today because of biological activity. Biological organisms, plants and bacteria that harvest the sunlight and make a lot of their molecules from that, and using CO2 as a input molecule, they produce oxygen. And so that's a real change in the Earth that came about by biological activity. But early on, it was not strongly oxidizing. Miller had hydrogen gas, H2, in his experiments, and would have had methane in there. And so these are what we call strongly reducing. And now people are, scientists that look at the early atmosphere are saying not so reducing, but I've heard people say, oh, well, maybe it was neutral. But then when I push on the, planetary people a little, they'll accept that maybe it was mildly reducing. And so there's kind of a whole spectrum. And I'm okay with that. If it's mildly reducing, we could still do some of the chemistry that Miller did.
And so just to review Miller's experiment, he not only had that atmosphere, those atmospheric conditions, but also was including lightning strikes as a way of inducing these reactions.
Right. Yeah. And that's an energy source that he has in there, because the molecules that he's making are not the lowest energy molecules, and to really get that reaction going and to make molecules that are actually, in a sense, a little higher energy state, you need to put that in. And so he was doing that in different ways. He did that with the electric discharge that you're talking about, was a model of lightning. He also did it with UV photons, that can produce some as well.
Just like the sun's energy.
Exactly. And so, that fits really well with a model for the chemistry on the early Earth, that we would have had this atmosphere that had these different gases in it, like ammonia, we would have had nitrogen, we would have had probably, I think, some hydrogen, maybe some methane, we would have, of course, water in there, and certainly plenty of CO2. And to really get the molecules of life, you do have to put some energy in there.
So we have a question from the audience. How do scientists figure out what the early atmosphere was like?
Oh, so there's some data that comes in from the geologists. The geologists have some minerals that were formed on the order of about 4 billion years ago. And so, of course, we don't have many rocks that are that old. And sometimes what we have are a rock that is 3.8 billion years old, but inside of it, there could be a small inclusion that they know is even older than that. And they're very clever. They can do this mineralogy, study the form of the minerals that are in these rocks. And from that, they can put constraints on what the atmosphere must have been like, because they can tell you that, oh, this type of mineral won't come out unless you have plenty of CO2. This type of mineral won't come out unless you have some dry land. This one only comes out as a sedimentation, so water on top of land. So there's things like that I think constrain us. But from there, clues come in from looking at the atmosphere of the other planets in our solar system, because they may have, be similar to that or have a starting point that's similar and didn't have life changes the way ours has. And then there's also theoreticians that will take say, different starting conditions and then run it forward and see how it progresses, and does it match with other atmospheres around planets, and would it also be consistent with the mineral record. We have to piece all those together, and certainly not, I would say, definitive in the end of what the atmosphere is like. But again, there's a whole spectrum, and we can kind of narrow it down.
So then how did the study of meteorites start to call into question some of this focus on amino acids that that Miller's experiment had brought to science? And many of us learned about Miller's experiment in our biology class, intro-bio classes, and it's a common textbook example.
Yeah, so I wouldn't say that it was in contradiction. In fact, in these meteorites, when they first fell and they were analyzed, they found that they contained a lot of the same molecules that Miller had found. So that was actually very reassuring. It did change the picture, though, some, because then we had another scenario that instead of all the molecules being made on the Earth, it's possible that they were being delivered from outside the Earth. So there's people that have swung all the way to saying that they think that the majority or most of the ingredients that gave rise to life fell in to Earth. And even today, we know that meteorites are coming down that have amino acids in them, and this would have been much more common early on. So it's very possible that a lot of material came from outside. I think it's also possible that some was made here, or maybe the majority was here. So I would say, if anything, it actually gave us more options rather than limiting our options. And by the way, I just add something that is, I think, very interesting about the meteorites story, which is that one of the meteorites that's been most influential in our thinking about prebiotic chemistry, that is the chemistry that gave rise to life, is one that's called the Murchison meteorite. And it fell in Australia in 1969. And what is so coincidental about that was that NASA had set up the laboratories to analyze the rocks that would come back from the moon to look for amino acids. And so when this meteorite fell on the Earth, NASA was right prepared to analyze molecules that had come from outside the Earth. And I think that that was just such a wonderful coincidence, because we got a wealth of information out of that.
Yeah, that's totally cool. Sometimes the timing is just right. Okay, so what are some of the things that you've explored about how water and temperature on early Earth could have affected RNA or Proto RNA, the earlier forms of polymers that were kind of RNA-like that might have led to RNA evolving?
So everybody, I think, would accept that water is necessary for life as we know it, right? Our bodies are mostly made up of water. Our cells, if you dry them out, they'll die. You got to have water. So we're all in agreement on that. And so most, I think, researchers that work on original life think that life started with water around. That's a useful, say, starting point. But one of the challenges is that our molecules that we have in our body, our polymers, like RNA being a polymer and proteins being polymers, they're actually not stable in water. Now, they're stable in us, and we can live because they're what we call kinetically stable. That is, they're not going to fall apart immediately. But if left long enough, they will actually break apart. And that's why our body's always making new ones and repairing them. You have to put in energy to make these molecules and have them be present in an aqueous environment with water around. And so in my lab, we've been investigating a very old hypothesis. It goes back probably 50 years or more. And that is the idea that the way the first big molecules, the polymers of life, were made is that they were on dry land that was intermittently wet because of rain, but then dry because the sun comes up and bakes the surface. In this very simple scenario that all of us see happening in our daily lives, we see rain come and puddles form, the sun comes up, it dries out. That's actually the type of environment that you would need to make these types of polymers that we find inside of us, inside our bodies today. You need a time, say, where it's favorable to give off water. And that's what would happen when amino acids react with each other to make a bond like you have in protein. But then you need a time when these molecules are active, because the molecules inside of us are active really in a wet state. And that's something you can get twice a day if you just, say, have a morning and night that coincides with rain and then drying by the sun. Or it might be something seasonal. It might be that during the summer it gets very hot and dry, and that bakes out maybe even a whole lake, but then it gets rehydrated in the winter. So we're far enough removed from the origin of life that we don't know really what the cycle time was for creating molecules. Maybe they were created on a daily basis, or maybe it was a yearly basis.
So I've read that you've jerry-rigged quite a few different contraptions for studying early Earth conditions and their effects on polymers. Can you describe some of them for us?
So we make what we like to call them day-night machines, but again, they could be reflecting seasonal variations. But what we're really doing in all of them is adding water in and then drying it out. And we have a number of different variations. One of the first ones we made was built around what's called a PCR machine. And PCR is actually, as you know, polymerase chain reaction. It's the reaction that is used to amplify DNA. It's used all the
creates
time.
Many copies of DNA.
Right. And interestingly, that in a PCR machine, what it really does is just swing back and forth and go to a hot temperature, a cool temperature, swings back and forth. But you have enzymes in there that respond to that, and they replicate the DNA that way. But we thought, oh, that type of machine is perfect for drying out samples. So as you heat it off, you'll dry off water. And so we took one of these PCR machines and we added to it another machine that would drop water back into test tubes. And so we could cycle this. We made that in the lab. That worked well. But then we decided we wanted to make a different type that didn't hold the type of tubes that we used in the lab, but actually could use kind of a flatter surface that could mimic, say, a surface of the Earth. And so we made one where it works with ceramic plates, like just a flat plate or plates that have indentations in them. And then we needed a method to dry these, and so we thought about all different ways to do this, and then we came up with the idea just to use a toaster oven, just to put them in there and dry them out. And actually it works really well. My students are still using it. We have some really nice data from that. And so we're right now planning the next generation of this. We're going to build another one this summer.
Toaster oven 2.0.
Yeah, well, this one actually it might work off a toaster oven. I'm not sure yet. It might work that or a PCR machine. But this one is going to have the capability that when the water is added, that it can mix it really well, that it can take it up and mix it around. And that might be a little harder to imagine from a prebiotic standpoint. I guess you could have rainstorms that are really hard that wash things off of rocks and that. But that's something that we found, is that mixing them up could change your results some. And so we want to really understand how much that affects the outcome. So we're going to make a machine that can do that.
Cool. So we have another question from the audience that kind of relates to your answer earlier. They ask, why would the drying cycle help with polymer building?
Okay, so there are two reasons that it helps. When we make a chemical reaction, say we're going to bring two amino acids together, we're going to make what we call an amide bond. For one thing is that that is not what we would call thermodynamically favored. Energetically, it's not favored. And you can write a very simple reaction equation where you say two amino acids come together, and what is produced is a dimer, we call it, or a dipeptide. They're joined together, and a water molecule. Now, if you're in water and you write out this equation, you're producing water, but you already have a lot of water there. So the reaction is actually favored to go back where the amino acids are apart, and they've actually taken up a molecule of water. So when we dry out the sample, we're driving it, as we say, to the conditions where those come together and water is produced. Also, when you're drying, you're bringing the two molecules close together. You're increasing the concentration as they dry out, and that will help the reaction. Another thing that will help the reaction is that in order for these molecules to come together and react, we actually have to add a little bit of heat energy to it to get them to jump over a barrier, as we call it. And that works quite well if we're in a dry state and we heat them up, then they'll come together. The problem is if we try to make that bond by heating them up while they're in water, then those molecules that we've already made the bonds with will actually break apart. So it really is good to dry them down and heat them up.
And so it really is Earth cycling that is, potentially incubating the polymers of life.
That's how I see it. I see it as we had these ingredients that were there on the Earth, and then the Earth spinning on its axis in front of the sun. I see that as like this engine, right, that was providing the driving force to get it all going.
So, so what are the steps that you've seen proposed, or that you think are feasible for the formation of amino acids to lead then to an organism, or the formation of RNA to then lead to an organism that's reproducing and expanding and diversifying?
So I think that there's some transitions or major events that occurred during the origin of life that are very common, or they were held in common for the different polymers we have in life. It would be the same for the peptides, it'd be the same for RNA, it would be the same for polysaccharides, as we call them, sugars linked together, and it would be the same for the membranes, which are really associations or kind of polymers of lipids, as we call them. And so I think that the stages were similar for each of them. The first stage is, of course, that the molecules that make them up were produced, the building blocks. You got to have that material. You can't get started without it. And so I think we had these reactions going on in the Earth that produced it. The second phase in this march toward life is, I think, that molecules that were similar to each other collected together. And the way that I think about that is that, when you have coffee rings, when you spill water out, you can, or spill some coffee, you see it dry, and you can actually see rings, and depending upon whether you had cream in there or not, you can actually see kind of variations in the color and that. And this tends to be because the molecules are separating out from each other. And I think that there must have been some process on the Earth that allowed sugars to collect with sugars and amino acids to collect with amino acids, because I think if these were being produced in the atmosphere or they were being delivered by meteorites, if they were all mixed up, it'd be really hard to get the chemistry going the way we need it. So I think that the first transition after making the molecules is getting them to kind of separate a little bit, and then we had these drying rehydration cycles, and we're making polymers. That's the next big step. And then I think that these started to, you might say, discover or acquire some catalytic activity that took them to the next level. And then the next one is that I think they started to cooperate with each other, that the peptides started to work with the nucleic acids, the RNA, for example, and that these started to work with membranes, they started to work with the polysaccharides. And in my opinion right now, that is what I would really call the start of life. I think that we could have a lot of polymers that were synthesized on the Earth, a lot of polymers that could catalyze reactions. But I think that the really big part is when polymers cooperated with each other. Because I like to think of us as living organisms as really just a cooperation of polymers. Right? We have RNA inside of us that is reading the code that's stored in the DNA, that is making the peptides, that are making the proteins. And then those proteins turn around and they make the nucleic acid, they make the RNA, and the cycle goes around and around. And they're all encompassed in this lipid membrane that needs the proteins to make them, right? So I think that there's this cooperation that is absolutely central to all of chemistry in life. And I think it was probably also the major chemical step that took us from what you might call non-life or pre-life to what we would recognize as our chemistry of life.
And so your lab has been able to make a sort of proto RNA molecule that's made up of 2 nucleic acids rather than 4. Can you describe how they did that and what it is, and how you think it fits into the potential story of the origin of life?
Sure. So 1 second, I'm going to grab a model.
Cool.
So just, I think it illustrates how close we are with the molecules that we have in life today. To actually clarify something, we haven't made a full proto-RNA polymer. What we've done is we've made some of the building blocks that you would need, like the monomers that could be stitched together. But to illustrate that, this molecule right here, or this model, this is adenine, or adenine, which actually is the A in AMP, and this is one of the bases that's found in RNA. That's an A. It turns out that if you take this molecule and you put it in a solution with ribose, which is the sugar that we have in RNA, which is supposed to connect right here, you can dry it down, you can heat it up, maybe you get 1-2% yield in the reaction. Only about 1 or 2% of this will be converted to the next step of what you need for RNA. And if you take the other bases that are found in RNA, the other 3, you don't get anything. They don't react. And so what we've been looking at is the possibility that some other molecules came before. And this molecule, has some similarities to adenine. This is called triaminopyrimidine. And this molecule is interesting because it could actually pair in the same way that RNA has base pairs with another molecule. And what we found is that if we take these molecules and we dry them down and heat them up with ribose, they react very well to make molecules that look like the building blocks of RNA. In fact, in one case it's 60% yield, and the other it's over 80% yield. These are great reactions. And then these molecules, you mix them together in water and they start pairing with each other and they're stacking up. And you don't get that with the monomers or the building blocks of RNA that you have in life today. So we can make just some very small changes. In fact, this one's called barbituric acid. And that one is so close to the molecule that we have in life today that if I just remove 1 atom there, if I change that from what was an oxygen to a hydrogen, I just switched out that 1 position right there, okay? The molecule that it was, barbituric acid, which you can make in a very nice model prebiotic reaction and reacts with ribose very nicely, I changed that 1 atom right there, and that's uracil. That's actually a molecule that is found in RNA. That's one of the bases in RNA. And so we're thinking that it's possible that the earliest RNA-like polymers were very close to RNA, just changing 1 atom here or there. So we'd still recognize the structures as we see them. But chemistry, one thing that makes it so exciting but also so challenging and it's hard is that you change 1 atom and the reactivity of the molecule can completely change.
And so let's see, I want to make sure I'm asking all the questions I want to ask.
Sure.
And so how would you see that as then leading into RNA being made, and then from there, the organisms that are membrane bound and beginning to have that cooperation between the polymers?
Well, what I think is that RNA and DNA that we have today, in many ways, they're just absolutely perfect. They are perfect for the functions that we need them for in the cells. And so what I think is that we started life out with what we would call a proto-RNA or a pre-RNA, something that came before RNA, an ancestral molecule. I think that molecule was more easily synthesized on the early Earth than RNA would be. But there was certainly much room for improvement. I think that the RNA that we have today probably makes better structures than these earlier molecules would have. I think for sure DNA is able to store information better than the early molecules could have. They probably wouldn't have been as stable. And so but in order to get those molecules to transition, say, from the earliest ones to the more advanced and to what we have today, we needed enzymes like we have in life today. We have enzymes, right? Like proteins that break apart the food we eat, but also stitch together the building blocks to make our proteins and all the materials that make up our bodies. We need these enzymes. And I think that was probably true early on, that these molecules would not have been able to progress from their earliest forms to the latter ones or to RNA until they themselves had actually produced catalysts that could make the subunits that they needed or catalyze the reactions that were needed for the next generation, so to speak, of molecule. But once they were able to do that, kind of tweak the molecule, experiment with some changes, when they happened upon a change that was beneficial, then that would have supplanted. It's just the way evolution that we see today. We know that we have the emergence of new species because, right, there would have been, say, mutations or just some natural variations that you have in genetics because of, right, the way our genomes work. But once you come up with a species that is better suited for its environment, it progresses, right? It's just natural selection. I think we would have had the same thing at the molecular level, that there would have been testing of different RNA-like molecules, and each time there was a molecule that was better suited for, say, information storage or for catalyzing reaction, it would have taken over.
So it's just like natural selection, then there's a variation of lots of different polymers, and then as they had an advantageous attribute that would have allowed for better cooperation and self-replication, then that would have been selected for.
Absolutely, and something that's key that you said there was about better cooperation, because I actually think that the chemistry to make RNA is hard enough that it may have required protein catalysts, enzymes. And if that's the case, then that would mean that we might not have had RNA in life until proteins were being made. And we know that RNA makes proteins today in life, and so it might have been something that an ancestor of RNA might have started making the protein, and then there was kind of a feedback between the protein and the nucleic acid. And it could be that it changed the nucleic acid into RNA because that was actually better for making the protein, right? And that the proteins were able to make the components of RNA. So I think it could actually go that deep, that the cooperation between polymers was so important and so powerful that they actually affected each other at their basic structure. But one thing that I like to say is that when we think about cooperation, it's not like you have, say, one molecule that can do 10 things well and another one that could do 10 well, and when you put them together, they can do 20 things. I don't think it's that way at all. I think it's that you have one molecule that can do 10 things, another that can do 10. You put them together and you can do 1,000 things or 1,000,000 things, right? Because it's synergy, right? And they really complement each other. And I'm thinking that that's how it was, that life really did not make this big push toward what we see as life today until we got this cooperation between polymers.
So we have another question from the audience. This is from Mark from the Cronin Group, Mark Garcia from the Cronin Group. He says, greetings from Glasgow. And he says, have you actually managed to get oligomers from the new nucleotides?
Oh, yeah, good question. No, we haven't. So we make these very nice, what we call non-covalent assemblies. And so if you think about the structure of DNA, where it's a double helix and in the middle the bases are all stacked on top of each other, our molecules come together in a very similar fashion, but that last bond that is between our units that are coming together to make them into covalent polymers, we haven't been able to get that to form yet. And a very timely question, because I was just up in my lab a couple of hours ago, and we were talking about the next round of experiments, the next types of chemistry that we're going to try to get that one last bond.
So that's what you're working on now, is on getting the oligomers from these
Very
nucleotides.
timely. Yeah, we're working on it. Even today, as we speak, my students are working on that.
And just for folks that aren't so biochemically inclined who might be listening in, what is the implications for that? What are these oligomers doing, and why are they important for the nucleotides to be able to?
Yeah, this is really a good question, because we don't, in my opinion, we don't currently have a reaction that produces RNA or something like RNA that is acceptable as a prebiotic reaction that could have gotten everything going. Now, if you look in the literature, there's been people that have said, well, this looks like we've got a way to make RNA. But there's a couple of problems with it. One is that typically in making these RNA molecules, they've had to use what we call chemical activation. They've had to make the molecule very reactive. And then we can get them to stitch together and make a polymer. But it's really hard to imagine how that activation would have happened on the Earth without the aid of enzymes, without the aid of energy harvesting systems. And so we have kind of a problem with that. The other thing is that if you look at the yields, if you look at how much of these molecules are being made, it's actually very small. It's just a few percent typically, and they tend to be very short. And so we're not satisfied yet. We're
like short-lived.
Short-lived, and also short in length on how long
Okay.
the polymers are. And that translates then to what they could do, what type of enzymes they could be, right? And so we're not, a lot of us are not satisfied yet in the field. We think that there must be some reactions out there that we just haven't discovered yet, working with molecules that aren't exactly RNA, but a little different, that will allow us to produce molecules easily, in high yields, and very long ones.
So I just want to ask a few more questions about just how you became interested, or how you ended up studying the origins of life, because it's not like your earliest research was not directly, it was related to DNA, but not directly related to the origins of life, right?
Yeah, exactly. In fact, the earliest research that I did when early on in graduate school, I was actually looking at proteins, another great polymer of life. But I was looking at proteins that bind to DNA, and that was really what my thesis work was about, studying those proteins. And the more that I studied this protein DNA complex, again, back to the idea about polymers cooperating, I was looking at these polymers that are working together. The more I studied this system, I became more and more intrigued with the nucleic acid part. I mean, the proteins were interesting, but the nucleic acids, I thought, wow, the type of functions these do, this information storage, I thought that's really intriguing. And then the more I looked at these molecules, I thought, this is really almost a bizarre molecule. I think that when we look at DNA, we've learned about it back in even grade school now. You learn about it in elementary school. And it's so familiar to us that I think we don't even think about the structure being a difficult structure to comprehend chemically, because we just see it all the time. But as a chemist, as I was looking at it, I thought, this is such a perfect molecule, but I have no idea how this would have come about. And so that was kind of this turning point late in my graduate school research, where I thought, wow, if there's one thing I'd like to understand in my career or my life, I'd like to know why this molecule was picked and how it was picked. And so from then on, even though I did a lot of other very basic research along the way, I kept thinking about this question about, where does RNA come from? Where does DNA come from? And so I really got obsessed, I'd say, with that question, and I wanted to answer it. I want to know the answer. And a lot of times what I also say to people is that, I'd like to be able to get the answer, but if somebody else gets it, that's great. I'd just like to be able to understand it, or I'd like to be in the room when they announce it, because it's such a good question.
So you spoke recently on the podcast Story Collider, and you told the story about your mother, who's a devout Catholic, and about how she didn't really understand why you were looking into the origins of life, and didn't really understand everything that you were looking at and researching. And so I just thought it was a great story, and wondering if you can just talk a little bit about how you got your mom and maybe some of the other people in your family, because I think pretty much all scientists, all of us, deal with having family that or friends that don't really understand what we're working on. And so how did you figure it out? How did you go about getting folks to appreciate what you found, what you had dedicated your life to?
Well, yeah, that's a good question, because it's a continuous process, I would say, with different family members. It is sometimes hard for some to appreciate why you want to study the origin of life. There are some that right away say, "Oh, that's a great question." Others that have such strong faith that they say, "Well, it's not even a question; it was done," right? Um, but I would say that when it comes to my mother, she didn't really have a problem with the idea that she's a devout Catholic. She, in a way, believes in creation, but she doesn't believe in it in a way of, just these 6 days. And when I would speak to her about my research and trying to understand how life was started, about evolution, she had no problem with her faith. To her, it was as if we were unraveling how this was done by the deity that she worships, right? Um, and I'm okay with that. I never tried to push her away from that. And I would say that one thing that I encourage all that in any conversation that I have with somebody that is very devout and religious, I respect their views in that. I'm not going to try to change their views. If they have questions for me and they're scientific, I answer their questions. And I think that's a good way to go about it. When I think about my mother, I respect her tremendously. A wonderful person, and I always say, I never be where I was today if I've succeeded in anything without her, because she was somebody that was at the same time, I would say, encouraging, but also forceful and achieve your full potential. She really drove us. And so I have this respect for her, so I'm not going to ever belittle anything that she believes. No way. I also have this feeling where she's been living, with her beliefs. She's 92 years old, right? Since the time that she can remember it, 4 or 5 years old. And who am I, after 80 some years of her believing that, to say, no, you shouldn't believe that? No way. I'm not going to get into that. So we actually have a fantastic relationship with each other. We talk very freely about almost everything.
And it was, in the story that you told, you talked about how you spoke at a seminary, and it was like she was finally, she finally saw some of the ways that you were describing it in her language. And was that something that you feel like was a turning point, or helped her see some of your work in a new way, or in a way that she understood?
Yeah, I would say that I wouldn't say it was a turning point, because I think that she had already gotten to some level of appreciation for the work I did. But I would say that it really went to another level, because at that seminary, they had invited me in because they had a series of lectures about science and religion. And I found out after I'd accepted the invitation that I was definitely on the furthest side of the science. They had people that were, I would say, more on the philosophy side and in the whole spectrum, but I was certainly the furthest on the science side, so it was challenging to relay this to seminarians and the others. It was an open audience; everybody could come to that. But what I just loved at the end of this, that when the Monsignor, who's in charge of everything, got up at the end, he just put it in terms of, of how wonderful God's creation is, like as if he had seen it from another perspective through the scientist's eyes, and it was just like another appreciation of it all to him. And I was okay with that. My mother was just delighted, that I could share something with the Monsignor that he appreciated.
So in addition to the work that you're doing on the oligomers, what are some of the other directions that your lab is going? What are you working on now?
So we're attacking really different parts of the origin of life problem. We're looking at not only the origin of the nucleic acids, we're looking at the origin of proteins or peptides. We're trying to understand that. We're trying to understand how other molecules that would have been present on the prebiotic Earth would have helped these molecules form. And this is like a whole nother dimension that I think of, because for I would say decades, most people that were studying the origin of life were concerned with what are the molecules that we have in life today, how would these have been formed on the Earth, and then how would these been stitched together to make the polymers of life and started functioning. But I'm not the only one. Others have thought before maybe life started with different molecules, but maybe also there were molecules that helped life get going, and they're not even in existence today. And so this is, like I said, like a whole other dimension. We can think about chemistry in terms of a space, and this is like a vast space, like an unexplored area for us. And we're having some success with it. One thing that we found is that if we take amino acids and we dry them and heat them, we can make some short peptides. And that's been something that I know that the Cronin group is actively working on in Glasgow, and there's some other groups that have worked on this in the past. But what we found is that if we take these other molecules called hydroxy acid, this whole class of molecules, and we put that in with amino acids, we can make the peptides at lower temperature, and it's easier reaction to get going. But what's interesting about hydroxy acids is that these are also formed in that reaction that Miller did when he produced amino acids. And they're also found in meteorites. And it looks like the same chemistry that makes amino acids also makes hydroxy acids. So it's almost hard to imagine that these two classes of molecules would have been present together on the early Earth. And even in life today, we have hydroxy acids inside of us. These are so similar to amino acids. It's really, again, just one atom is different. On the amino acids, you have a nitrogen. Hydroxy acids, you have an oxygen. Very similar. And in fact, life even uses some hydroxy acids to make its amino acids. And so we were surprised that nobody had tried this before, and we were also surprised about how easy the reaction is, how much better it is if we have the mixture. And so we're doing that with the peptide synthesis, but we're also looking at molecules that were possibly present on the prebiotic Earth that could have helped the nucleic acids form. So in a way, it's challenging because we have to think about many more molecules. It's also challenging because you could think that the reactions are getting messier because there's more molecules present. But in that mess, there could be a lot of reactions that we would be missing if we just use the pristine, say, isolated chemistry that we have in life and didn't consider what also was possible at the beginning.
Well, it's fascinating work. I want to begin to wrap up, and so do you have any points or comments or things that you'd like to talk about before we have just 5 minutes left? So
sure. I'll bring up something. Some people ask me, why do you study the origin of life? And in fact, I would say that scientists tend to fall into two categories when it comes to the origin of life. There are those that are the origin of life enthusiasts. And sometimes I like to call them the closet origin of life researchers. And that is because sometimes I'll be talking to a scientist who has never worked on the origin of life or never published on it. And maybe they don't even know what I work on. But then when I tell them that I'm working on it, then they'll get really excited and they'll say, oh, I have this idea. I've been thinking about this. Maybe this is relevant to the origin of life. And they just kind of really light up about it. And so that's why I like to call them kind of the closet origin of life researchers. They're ready to come out, right away. There's them. And of course, those of us that work on us are kind of used to be that way. And now we're really out in the open talking about what we do. And then there's another class of scientists that sometimes say, well, you shouldn't study the origin of life because you can never prove that you're right. You can't go back in time, right? And so to them, sometimes they say it's kind of a futile exercise. And in that way, I think they're not adventurous enough. I think that we can get to a point where the chemistry that we discover that could produce lifelike molecules may become so compelling that it's kind of hard not to accept that this is the way it happened. I think we can get to that. But I also think even if we fall short, I feel that as scientists, there's this gap in human knowledge. If we don't even have one possible scenario for the chemical origins of life, we're kind of missing out. And I think that if we can provide one scenario, maybe it's not the only scenario, maybe it's not even the scenario that happened, but one plausible chemical system where we say these molecules were most likely present on the Earth, we let it undergo these type of changes in the environment, and we produce molecules that look very much like life, and they start to evolve. That, I think, is going to be really a shift in people's thinking about the type of chemistry that's necessary for life, how widespread life may be in the universe. And I think it's also going to show how far we've come as chemists and biologists, right, and physicists and geologists, where we can put together such a scenario. I think that will really be something that changes people's view of how much we can understand about life.
And with so much of basic science, which just comes out of a sense of curiosity, then you never know what kinds of applications can come out of that. And that's how most of major breakthroughs have happened in science.
Absolutely. I would say that it's not an understatement to say that in chemistry we have not made molecules that can do what living organisms can do with chemistry. They can evolve molecules like nothing else. And if we can come up with a system that allows us to take very simple molecules and produce molecules like RNA, I think it's also going to be in the same type of chemistry that we find a way to do evolution with a non-biological system. I think when we get to that point, we're going to be able to present something to the chemistry community that's going to allow us to evolve new catalysts, new materials, new therapeutics. I really think things that were not possible before will be possible based upon this chemistry.
Well, it's fascinating research. Thank you so much for taking the time to explain it to our audience and to me. And good luck with it. Good luck with all of your research and setting up new ways of studying it.
Well, you're very welcome, and thank you for having me on. It was fun talking to you.
All right. Good luck. Thanks, Nick.
Oh.
Bye-bye.
Bye-bye.